So, in this talk, I'm gonna look at certain, add a little more to what you've already learned about the impacts of cover crops in the vineyards, and how these cover crops impact the communities of arthropods in the vineyards and insects in the vineyards. Well, we wanted to see whether we could see more of the benign insects in the vineyards and how some unwanted insects behave or are present in the vineyards. Of course, it's the insect populations have been decreasing for a long time. This is a major problem in every vineyard, and this has an adverse impact on the ecosystem services. There are less and less pollinators, and there are more and more unwanted bugs. There are various invasive species of insects coming to the vineyards, and they cause real phytosanitary problems. Erosion is another issue. Sometimes we have these huge downpours, sometimes a year's precipitations. Vineyards get in 2 days, and they just flow the earth down the hillsides. And this results in a deterioration of soils, and they lose their ability to retain water. And also climate change and extreme weather conditions are also challenges that we need to face and respond to some extent. And of course, another objective is to reduce the pesticides that can be used. This poses another new challenge to farmers. So how can we mitigate all these problems? How can we be more resilient and ready to respond to these challenges? On the left-hand side, you can see what it looks like when a landscape looks like when you have intensive agriculture. Normally, monoculture is present here, same species of crops, and no any sign of natural vegetation, while in the right-hand side, you can see a landscape using agroecological practices, and you see there are lots of green corridors between the fields, and there are small habitats, and there are different crops grown in different smaller sites. And most ecologists today agree that if you bring life back into a landscape like this, you can increase the number of useful insects much more efficiently. Of course, everyone is afraid that if you leave some trees nearby your fields, then the unwanted bugs will come from there and propagate there. But normally, and lots of research has been done into this, benign insects normally grow in population much more than the unwanted insects do. And so this results in a much more diverse ecosystem, which is much more resistant to alien invasive species, for instance, if you have such an agricultural matrix. My colleague Tomáš already mentioned results from this Lifeline project. We have 3 sites, vineyards in the Tokaj area and 3 in the Eger area, and 4 alleys have been sown there with the cover crops, with the 6 species mixture and the 19 species mixture, and then we have a control alleys in the vineyards where we leave the natural vegetation growing, and we selected insects from these sites And this is what the cover crops look like in one of the Eger sites. This is growing a little too high. Normally it's lower, but and normally they clean the cover crops from under the vines. Of course, it's only in between the vines, the rows of vines. So I brought along some samples we took in 2023. We used sweep nets to collect a sample of insects 5 times every season in 5 different months. And in early July, we also checked 75 leaves randomly, and we were mostly looking for American grapevine leafhopper. We tried to identify the number of American grapevine leafhoppers in the vineyards, but also looked at the possibility of other insects being present on the leaves. And we also did some phytosanitary tests as well, 2 to 3 times per season, normally in August and September. We went through all the and we looked at parts of the grapevine, and we looked at the symptoms of various grape diseases here. So let's look at what we found in between the rows of vines. This dark purple color shows dark magenta color shows the control sites, and this green color shows the low diversity cover crops, and the yellow means the high diversity mixture cover crop areas. And you could see that both in the 6 species alleys and the low diversity mixture, the number of spiders were higher in both. We also looked at the numbers of cicadas in there, and there was not a major difference in terms of the number of cicadas we found. So the high diversity and the low diversity mixtures showed similar values, but it was close to what we could find in the control sites. When it comes to parasitic wasps, which can actually kill some unwanted and adverse bugs like mites and moths, and that was a high number of parasitic wasps in the vineyards where we sold the mixture of cover crops there, and in the control site we could find much less of them. And when it comes to the various Cyrophoidea species, here the number of such species, these flies were also present in higher numbers in the sites with cover crops. Particularly, we could find a high number of these where we planted 6 species of cover crops. And it looks like that normally we have a higher number of benign bugs in the vineyards where cover crops are used. And if you look at the Apoidea species like wasps and bees, then you see the same. Left-hand side you can see the species and the specific species average, and on the right-hand side you can see the average numbers of species. And also there are twice as many different species of bees and wasps you can find in the vineyards. Not only the total numbers of these species are higher in vineyards where you have cover crops. If you look at what you can find on the vines themselves, on the grapevines themselves, if you look at the number of spiders, you can see maybe a little more more spiders you can find if you have cover crops in the vineyards, but that is not a major statistical difference compared to the control areas. However, the grain lacewing eggs you can see, you use these nice, these stalked eggs. This is what the grain lacewing lays on the grapevine leaves. And these are predatory species, and the low diversity mixture cover crops we could find in these sites where we use these low diversity mixture cover crops. We could find much more green lacewing eggs in the vines itself, in the canopy of the grapevines. It was like 50% more than in the control site. If you look at the American grapevine leafhopper on the left-hand side here, then you can see that there was less of them we could detect in the low diversity cover crop areas. In the 19 species cover crop areas, we could see a much higher number, but in which is just a little more, not significantly higher than in the control area. But this is what we could see in both the green lacewing eggs, and also the probably if you use the right mixture of species in your cover crops, then you can increase the number of certain unwanted species, decrease the number of certain unwanted species in the insect species in the vineyards, and there will be less cicadas in the and grapevine leafhoppers in the vineyard, because they have these vector activities, you know. Let's look at different fungal diseases. On the left-hand side, you can see how we looked at the 100 grapevine pots, and here on the left-hand side, we can see how many powder mildew symptoms we found on the leaves and how many in the clusters on the right-hand side. In 2023, there were very minor differences between, there were no major powder mildew issues. It was less than 1% the presence of powder mildew, and the treatment was not different in any of these sites. Now we looked at symptoms of black rot as well. The differences were only anywhere between 1 to 1.5% between the different areas. No major differences there. However, in 2023 was a very good year, and at least 2024 was a year where we could really measure downy mildew levels. Of course, the wineries were not very happy with the presence of downy mildew in the vineyards, but we were happy because we could do some tests there, and it was 15 to 20% presence of downy mildew symptoms in that year on the leaves. And it looks like that the lowest in the areas with the 6 species cover crops, it was like 6% less. The 19 species cover crop areas, it was little less occurrence of downy mildew symptoms, but it was a little higher. And normally, the biological activity on the soil at the soil level will be higher, and downy mildew cannot find any nutrients that it could use to survive the winter, and this may result in lower downy mildew occurrence in these sites. That's the explanation behind this. Here, this slide shows some test results. Phytosanitary test results from the previous 3 years, and generally the control and the low diversity is what you can see here. So you see the differences, and you look at the bottom line here, you can see that practically when it comes to, you see the powder mildew, black rot, and botrytis presence, and here you don't need to be afraid of cover crops, really, that you will have do more pest control as a result. This is not really cover crops doesn't bring in more fungal diseases to your vineyards. Really, there's no real change. So from a phytosanitary perspective, cover crops do not represent any sort of risk. You see that downy mildew occurs less often, on the average 4% less if you have cover crops, both on the spotted grapevine moth never caused a problem at all, and also grapevine leaf mites also rarely occurred. So luckily, no phytosanitary problems resulting from the presence of cover crops. However, lots of benign insects were present in the vineyards at the same time. So it looks like that cover crops seem to be a very safe way of improving diversity in the vineyards. So basically, to sum it all up, the population of various pollinating arthropods and other insects can be increased using cover crops. The number of wild bees will be higher in cover crop vineyards, and the number of different species of wild bees will be different too. However, the higher diversity cover crops will not help increase the number of such insects in the vineyards more than the low diversity cover crops. And the number of predatory species can also increase if you have flowering plants in the alleys between the vines. I'd like to thank you all for your attention, and thank all the participating or collaborating wineries to allow us to do these tests in their vineyards. Thank you.